So let's talk about a good controlled experiment. This is kind of piggybacking on what we were talking about before when we were talking about the scientific method, good experimental design, all those fun topics. But now let's get into the actual experiment. If you're creating an experiment, if you're running a lab, if you're trying to work through what it looks like to do higher level work like thesis and dissertation work, it all follows the same general guidelines. So first and foremost, if you remember back to that good old scientific method, it always starts with observing a problem, and following that problem observation, you are going to form a hypothesis. Now, a hypothesis is what you think is going to happen based on your prior knowledge and your research and your work done up to this point. It's what you think is going to happen when you set up the experiment and you run the experiment. And so most of the time when we're talking about a good hypothesis, one of the very first things we want to make sure we observe is that the hypothesis is testable, right? Sometimes you'll hear this referred to as a falsifiable hypothesis. It must be testable. It must be something that you can run an experiment on and actually get some type of answer to whether your hypothesis was supported or not. This can come in a couple different forms. So when you were in grade school, it might have been something posed as an if-then question, or sometimes you'll even see it asked as a question. But truthfully, when we're talking about a good scientific hypothesis, it should be a statement. It shouldn't really be a question. You should be saying this is going to happen. It should be something very specific. And occasionally, it is something that is going to require more than just a single set of tests or experiments to actually refute or support. But it needs to be based on past research. It needs to be based on something. It's not just a guess, right? This is something that is actually, you've done some work, you know some background info, and now you are setting up what you think is going to happen when you run the experiment. Also keep in mind that a hypothesis isn't going to mean much unless you have a good controlled experiment. And so I always think of it from the standpoint when you observe your problem, you come up with a testable hypothesis, you then state your null hypothesis, which again, that is what happens in the experiment if what you do, your independent variable, has no effect. And if you're not sure what that null hypothesis is, there is another video that you can follow that I did earlier that kind of walks you through how to do a null hypothesis and why it's important. Once you have that hypothesis and that null hypothesis, now it's time to set up the experiment. And keep in mind, when you are setting up an experiment, you have to make sure you are doing it in a way that you are controlling all the different parts of the experiment except that one variable that you are testing. We call that the experimental variable, or probably more commonly you hear it referred to as the independent variable. Think of it as it is kind of separate from the norm. It's independent from it. It is what you are changing. It is what you are doing in the experiment. And if done properly, you will only have one single independent variable, one thing that you are controlling the change on. And then you're going to keep everything else, and I mean everything else, exactly the same, like exactly the same, right? Are we making sure that we have something to compare to? And so a lot of times in an experiment, especially a product test or a medical test experiment, we will have the norm, what we call the control setup, and then we have the experimental set, which is the thing that has the independent variable, the thing that you are changing. And if we're really keeping everything exactly the same, a lot of times that requires the production of something called a placebo. And the placebo is going to be something that is going to give you something to compare back to, where you don't even know if the independent variable or the placebo is the one being tested. So it makes it so you are fully eliminating your bias or anything your expectation might be from that experimental setup. And if it's a true experiment, your dependent variable, your data should be coming directly from the observations from the experiment. So again, if you are setting it up properly, there's only one thing that is being changed, your independent variable, from the start setup to the end setup. And then your dependent variable, your data, is going to be what you are pulling that should actually tell you whether or not your independent variable is actually supported or not. And so in this example here, we're talking about a plant that is being grown with some type of fertilizer, right? And so we've got our control group over here. The control group, you may notice, looks exactly like my experimental group. So my experimental group, I've kept the same light, same temperature, same water, same type of plant, same container, hopefully even the same room and the same windowsill, right? The only thing that I'm changing between the original group and the experimental group, the control setup and the experimental setup, is going to be the application of some type of fertilizer. If that's the case, and if I do it properly, then I know that my hypothesis, let's say, was that the plant with the fertilizer would grow taller. It's something that I know I can collect data from. I'm only testing one thing if the fertilizer is present or not. And my null hypothesis for that type of experiment would simply be that the plants would grow the exact same height regardless of the fertilizer. So me putting the fertilizer has no actual effect. And that eventually gets us to the point where we are just testing that one single thing. Our dependent variable is going to be the plant growth, how tall the plant is. And if we did it right, everything should be kept exactly the same. Lastly, when you get to the very end of the experiment, you got to set up your conclusion. So a conclusion is pretty simple. It should always be directed specifically at your hypothesis. So most conclusions are going to start by restating your hypothesis, that thing that you thought was going to happen, that you made as a statement at the start of the experiment. And then you are just simply going to say whether or not the data supported your hypothesis. Also, if you have a null hypothesis, which you probably should, you should bring up that null hypothesis and you should say whether it was supported or refuted by the data. If your hypothesis was supported, fantastic. Probably going to mean your null hypothesis was not, and that's totally okay. Sometimes the null hypothesis is wrong and the hypothesis was wrong, and that's totally okay too. It just might mean we got to go back and do a little bit more research. In your conclusion, you should also have a closing statement type setup where you're actually talking about any important key takeaways from the experiment or from the data. I always think of this as like a relevant section where you're saying like why you did what you did and found what you found. You want to have an error section, and an error section is kind of the name, kind of self-explanatory, right? Anything that went wrong, you'd want to have listed in the error section, but you also want to have things that might have gone wrong. So things that maybe you didn't witness happen, but could have been a problem. So a lot of times in experimental setup, maybe you're not there to watch every moment of the experimental design. And so while you were not in the room, something could have happened to your setup, which could have altered the results. You'd want to include something like that. Maybe part of the actual lab design could have been wrong. So maybe if I'm doing that plant growth lab, maybe, the dirt mediums weren't the same, or maybe some of the seeds that I used in one of the setups were faulty, right? You'd want to basically kind of explore anything that could have gone wrong and certainly list anything that did go wrong. And then you're going to end with a little portion called the further study section, which further study is really just the term that we use for like thinking of now that you know what you know, what could you do differently? If you could make a new hypothesis, you could add to this experiment or change the experiment, what would that look like? In most further study sections, they usually have something in there where they ask you to state a couple of questions that could be answered in that further study. Kind of think of it almost like this is the next step, maybe even the imaginary next step for my experiment. So the whole goal for all of the conclusions is just to summarize what you did, state whether the hypothesis and the null hypothesis were supported or refuted, talk about whether it's relevant, the information that you had, any errors that could occur, and then also that further study section is important. And so just keep in mind when you're restating your hypothesis, when you're bringing up that final parts in that conclusion, you should always start with that. Your experiment started with you observing the problem and forming the hypothesis, and you want to make sure that your conclusion ends exactly where you started, right? Any final thoughts or insight are always a great thing to have too. Summarize any key points that your data actually found. But when it's all said and done, the conclusion's main goal is to make sure that it is specifically saying whether or not the hypothesis was supported or not.